Welcome, everyone. We are so delighted to welcome Dr. Zoe Donaldson to Emory and to the Center for Mind, Brain, and Culture. Dr. Donaldson is an assistant professor in the departments of molecular, cellular, developmental biology, and psychology neuroscience at the University of Colorado, Boulder. She received her PhD in neuroscience here at Emory and completed a postdoctoral fellowship at Columbia University before she joined the faculty at Colorado. Dr. Donaldson's research investigates the neural and genetic basis of complex social behaviors. Her lab combines perspectives from evolutionary biology, genetics, neurobiology, and psychiatry to uncover the biological mechanisms that contribute to species and individual differences in behavior. The goal of her work, investigating this socio-behavioral diversity, is to better understand diseases characterized by deficits in social behavior. In addition to publishing an impressive body of work in the top journals in the field, including PNAS, Scientific Reports in Science, Dr. Donaldson has received a number of awards and grants, including an NSF Career Award and an NIH Director's New Innovator Award. And please join me in welcoming Dr. Donaldson, who will be talking about neurobiology of love and loss, from genes to behavior to brain and behavior. Yes, thank you.
Thank you so much for that kind introduction. As mentioned, I actually did my PhD here at Emory University with Larry Young. So here's some throwback pictures for you guys. I definitely did not put a few pictures on here that maybe look even more antiquated and include current faculty. I am so honored to be back. There's something truly special about being asked to come back and give a presentation of your work at the place where you did your graduate career. And Emory was an absolutely fantastic experience. Larry, my advisor, once said that the second most important person in your life will be your graduate advisor, especially during the years you're doing your PhD. And he's not even here today, but I know that I can count on him to continue to write 4 million letters of recommendation throughout my career. So I want to transition now to what I've been doing for the past about 5.5 years. So I know that this audience is likely familiar with prairie voles, but I, like Larry, study prairie voles, and my laboratory is broadly interested in examining the biological basis of pair bonding and loss. And we do this from a multi-level perspective. So we look at everything from genes to brain and behavior. And so I wanted to just give a very brief overview of what my lab is working on, and then focus on one specific story today. So everything that we do in my laboratory is really fundamentally about understanding behavior, which means that we need novel ways to be able to investigate behaviors that are relevant to pair bonding and loss. And I'll talk about a few of those today, but ultimately we couple our assessment of behavior with a variety of different neurotechnologies that allow us to ask questions about the neuromodulatory control of bonding and loss, the neuronal encoding of affiliation and attachment. And in a new line of research, we become sort of increasingly interested in the dynamics that occur across two brains when individuals are interacting with each other. And I'll just sort of highlight this very, very briefly at the end. All of these are observational approaches, so we actually complement these with optical and genetic tools that allow us to go in and test our hypotheses by attempting to either break or make bonds through approaches such as optogenetics, which allows us to manipulate neural activity, or through CRISPR-Cas mediated functional genetic approaches. The story that I'm going to talk about today is one that I consider sort of a fundamental basis of understanding the biology of pair bonding and loss, and that's its transcriptional basis. So what are the gene expression changes that occur as a function of forming a bond, and what happens to those after you've lost your partner? So today's talk is first going to focus on how we've gone about identifying sort of the transcriptional basis of pair bonding. I'm then going to spend a bit more time talking about how we can actually leverage what we know about the biology of bonding in order to better understand loss. And finally, I'll finish by highlighting just 2 slides that have some of our future research priorities that I'm particularly excited about. So as you likely know, pair bonding is a fundamentally human behavior. It's fascinating. If it wasn't, we wouldn't insist on remaking Romeo and Juliet over and over again. It is innate. We do not choose to fall in love. It is complex. Anybody who's gotten married and has kids knows that relationships change as a function of experience. And we also know that the strong bond that we form with our partner ultimately serves to improve our health and well-being, but the loss of that bond is incredibly detrimental. And so I'm really motivated to study these behaviors for all of these reasons. And there are great reasons to study this behavior in humans. We have a complex vocabulary that allows us to articulate these really refined emotions that accompany pair bonding. But where we fall short is being able to look at some of the most basic molecular and cellular and circuit levels of pair bonding in humans. So what that means is that we need another species that we can use as a proxy that essentially demonstrates similar behaviors to us as humans. And so one of the most frequent questions I get, although maybe not from this audience, is why don't we study mice? And the answer is very simple. Mice don't form pair bonds. So while there are fantastic genetic tools within this species, we can't study something that doesn't exist. And so instead, in my laboratory, we take advantage of the monogamous prairie vole, which forms pair bonds both in the wild and in the laboratory. And because of their monogamous mating system, we actually have a whole suite of behaviors, complex social behaviors that are highly relevant to humans and are not displayed by our traditional mouse and rat models, making these guys an excellent model for studying the basis, the biological basis of these behaviors at every level, from genes to brain and behavior. So you might be asking yourself, how do you take a complex construct like monogamy or even pair bonding and distill it down into something that's quantifiable in a laboratory setting? And so for this, we rely on something that's called the partner preference test. And so this video here essentially shows you one animal in the middle that can freely roam, and then two animals that are tethered on either side. And in this test, this animal is going to go over first, and it's going to interact with a novel opposite sex stranger. And in this zoom in, you can see that this is a rather antagonistic interaction. Now, this is very common. When prairie voles form pair bonds, they only want to hang out with other prairie voles. And so here you can see that interaction with the partner, which looks pretty dramatically different. We call this huddling behavior, presumably because calling it cuddling would be a little too anthropomorphic. So if we take a prairie vole and it's sexually naive, it's not formed a pair bond, and we basically give it the choice between two opposite sex partners, what we'll see is that at a group level, they don't distinguish between these two potential mating partners. However, if we then randomly assign one to become their pair bonded partner, and in my lab, it's all arranged marriages, then what we see is after cohabitation and mating, we see this robust preference emerging to spend time with the partner compared to the novel individual. So this is called a partner preference, and it is what we use as a proxy for whether or not the animals have a pair bond. So the first question that we wanted to ask is just what are the gene expression changes that contribute to this change in behavior? And so in thinking about this, we sort of thought broadly about what happens when an animal forms a pair bond. And one way that we can conceptualize this is essentially to say that we are studying a shift in affiliative behavior state. So in sexually naive voles, they're generally affiliative to other species members, including other same-sex individuals. They may still be living at home with their family members, but once they find that special someone and they form a pair bond, now they are exclusively affiliative towards that individual. And so we know a few things. We know that this formation of a pair bond is not instantaneous. And in fact, work from my lab has shown that pair bond strength actually continues to grow as the bond matures over a series of weeks. It's also long lasting. So in data that I'll show you today, we can actually separate partners for 4 weeks and they still remember and demonstrate a preference for each other. And finally, this behavior, sort of long lasting nature, are underpinned by changes in biology. And so these are the changes that we're really interested in understanding. So we have sort of a behavior that emerges as a function of pair bonding. And the question becomes, what are the underlying sort of neurobiological basis for this? And so the first question that we could ask is, where do we expect to see changes in the brain that would support this emergence of partner preference? And for this, we lean on a converging literature as well as our own work that suggests that the nucleus accumbens is particularly important for being able to form and maintain bonds. So in humans, if you put people into an fMRI scanner, when they think that they're holding hands with their romantic partner compared to when they're holding hands with someone that they don't know, you see enhanced activation of the nucleus accumbens. And work that had its genesis here at Emory, we also know that neuromodulatory signaling in the form of oxytocin and dopamine is required within this brain region in order to be able to form a pair bond. And in work that my lab published in 2020, we showed that changes in neural encoding within this brain region accompany bond maturation, suggesting that plasticity within this brain region may be particularly important for underpinning the relevant behavioral changes that maintain a bond over time. So that for this reason, we decided to focus on the nucleus accumbens, but my graduate student, Julie Cedino, decided that she wanted to understand what might be changing at the level of gene expression within this brain region, leading to these changes in neural encoding and behavior. And she decided to test the hypothesis that pair bonding, like other changes in behavioral state, is ultimately mediated by a shift in transcription. So this has been studied in other changes in behavioral state, such as changes in dominance hierarchy or changes in where you are in the division of labor if you're a honeybee, but had never been explicitly looked at in the context of pair bonding. So go about doing this? Well, all gene expression studies ultimately identify genes as being up or down regulated relative to a comparison group. And so we took advantage of this sort of shift in behavioral state where we basically said we have sexually naive animals that we can co-house with a same-sex individual. They're affiliative towards that individual, and we can consider this to be sort of a control behavioral state. We can then take our animals that are paired with an opposite sex partner, and we can compare these back to our control behavioral state, essentially asking the question of what changes when an animal forms a bond. Now, I'm going to show you a number of plots that look like this today. This is called a volcano plot, presumably because these look like the plumes of a volcano. And what you see here, every single dot is a gene. And if it's purple, it means it's upregulated in pair bonded animals relative to their same sex counterparts. And if it's green, it means it's downregulated in the pair bonded animals relative to their same sex counterparts. And just to decipher these legends for you, on the bottom is showing you the magnitude of the change. How much did it change in expression? And on the y-axis here is the statistical significance of that change. Now, I just really want to hammer home that in doing it this way, we are defining the pair bond as opposite sex pairing versus same sex pairing. Now, this doesn't mean that there aren't interesting things to study with respect to what happens in same sex relationships, but it allows us a way to ask what is unique to these opposite sex pair bonds. So in order to do this, we were very interested in whether there was sort of a shift in transcription that was long lasting. And so we weren't so interested in this initial formation of a bond, but rather what are the long lasting changes in the brain that may maintain these bonds over time. And so what we did is we took male prairie voles and we either paired them with another male or with a female for 2 weeks. We then did our partner preference test, which showed us that they did indeed have bonds. And then we put them back with that partner and we harvested the nucleus accumbens tissue either 48 hours or 4 weeks later. We can then take that tissue, perform RNA sequencing, and we can identify genes that are either up or down regulated in our pair bonded animals relative to the same sex controls. And so we have these nice volcano plots, but the real question we wanted to ask is, we see these patterns of transcriptional change, but are the same genes being up and down regulated at both time points? So one way that we can address this question is to use something called a Morpheus plot. And this takes advantage of the fact that every single dot here has a value that's indicative of the magnitude of the change. So we can array our genes in a linear fashion from the most down-regulated in the darkest green there to the most up-regulated in the darkest purple. Now, if we keep our gene order the same, we can then apply a color in a second data set to tell us how similar these things are. And so when we do that and we keep the gene order the same here, what you see is a strong enrichment of purple at this end and green at this end, which tells us that genes that are upregulated in the short-term pair bonded animals, so those that have been together for a little over 2 weeks, are still upregulated in animals that have remained paired for 6 weeks. And the corresponding sort of downregulation is also true. So we actually have shown this about 4 different ways using 4 different analytical approaches, and it's on bioRxiv. But for today, I'm going to suffice to say that all of these analyses have shown us that pair bonding leads to remarkably persistent and consistent changes in gene expression. So in other words, there's a bunch of genes that change their expression when you form a pair bond. And as long as that pair bond remains intact, those changes in gene expression remain stable over time. So because these patterns of transcription are so similar across time points, what we decided to do was collapse them into one data set so that we have a very well-powered analysis here. where then what we can do is we can actually leverage this and say, based on the gene expression changes, what do we expect to be changing at the level of biology? So we can use a bioinformatic approach known as gene ontology that basically predicts certain processes to be involved based on these gene expression changes. So when we do this, we get a whole block of text that looks like this. Don't worry. The main point here is that if you focus in on these red arrows, about half of these terms are actually implicating glia, gliogenesis, oligodendrogenesis, myelination. So basically a really important role potentially for oligodendrocytes. This was a bit surprising to us. This was not where we were expecting to go initially. But when we did some digging, this actually makes a lot of sense. There's an emerging literature suggesting that myelination of axons is crucial for some forms of complex learning and long-term memory. And social bonding absolutely takes advantage of complex learning and long-term memory. Now, we can also dig a little bit deeper and we can ask the question of what are the genes that are actually contributing to the prediction of gliogenesis? And when we do this, we see sort of our most likely characters. So OLIG1, OLIG2, myelin basic protein, and GFAP are basically proteins that we know are expressed in glia. But what was a little bit surprising and really reassuring to us is this last one here. ERBB3 is a receptor tyrosine kinase. It binds neuregulin, and it's notable because it was identified as a potential candidate gene implicated in the evolution of monogamy across diverse animal lineages. So, found changes in behavior that occur when animals form a bond. And now we've delineated some of the molecular changes within the nucleus accumbens that might be responsible for these long-lasting changes in behavior. And then we sort of stepped back and we said, well, as humans, we're capable of forming bonds, but it's not that uncommon for us to form a bond, lose a bond, and form another bond. So this process is somewhat malleable. And so we wanted to ask the question of, having identified these behavioral and transcriptional phenotypes, what happens to them after a vole loses their partner? So in the next part of the talk, I'm going to talk about actually leverage pair bonding to be able to study loss. And I want to take a step back for a minute because there's some really compelling reasons to study loss. So loss is a central part of the human experience. We will all experience it at some point in our life, and it is profoundly bad for our health. In a subset of individuals who are grieving, they don't adapt to the loss, and they sort of wind up in this very severe and perseverative state that's associated with what's called complicated grief. NIH, despite how few social neuroscientists are studying loss, does have an interest in understanding it. And they actually define loss as a state of deprivation from a motivationally significant person, thing, or situation. Now, our work is definitely most relevant to this first category, but I hope that what we begin to understand will also provide some insight into these other situations of loss. Now, I want to take a minute and also just clarify that loss and its emotional manifestation as grief is not the same thing as depression. So colloquially, we have a tendency to conflate these two things, but they're not the same. This is evident in the fact that when we lose someone, grief is characterized by a yearning for that particular individual. Yearning is not a feature of depression. And furthermore, traditional sort of behavioral and pharmacotherapies that are efficacious for treating depression do not reduce yearning as this core phenotype. Now, we do eventually, most of us, recover from loss, and we think of this recovery as an adaptive process. It takes time. You have to relearn to navigate your world with the absence of this very salient and motivationally significant person no longer there. So what does that process look like? And I think this is the key question for being able to begin to design new therapeutics in order to address complicated grief, which we conceptualize as a stalling of these normal adaptive processes. So what I'm going to show you is our attempt to try to leverage the biology of bonding in order to study loss adaptation in prairie voles. But this is an inherently challenging question. How do you study something that's no longer there? And what we've come up with is, first of all, that we can operationalize loss in prairie voles via partner separation. And then we can look at the behavioral trajectory of an animal following loss to get a sense of when they have adapted. And having delineated that trajectory, we can then ask the basic question of what's happening in the brain across that same time course. So I do want to highlight that voles are not humans. So while we typically know something about the finality of the loss, partner separation is actually a very ethologically relevant model for voles. And this is because the most likely way that they lose their partner in the wild is that partner leaves the burrow to go find something to eat and instead gets eaten themselves. So we call these guys the popcorn of the prairie because they get eaten by quite literally everything that's carnivorous and omnivorous. In the wild, their lifespan is something like 70 days on average, and that's just because of heavy rates of predation. In addition, there's face validity to using partner separation as a model for studying loss, because we know that if we separate prairie voles from their partner, they exhibit a series of physiological and behavioral changes that mirror those we see in people that are acutely grieving. Now, I think this is a really strong evidence that we can study this process in voles, but we're not necessarily interested in sort of these acutely traumatic responses to partner separation. What we want to know is, can a vole adapt, and what does that look like? So the first question is, how do we know if a vole has adapted to the loss of their partner? And to answer this question, we sort of took two separate approaches. So in the first one here, we asked, how long does a bond last after we separate these guys from their partner? And then in a second question, we asked, when can a vole form a new bond with a new partner? We decided to focus on social behavior because this is the core of what makes social bonding and loss a unique construct compared to other forms of trauma. Loss is trauma, pair bonding is not usually trauma. Okay, so first question is, how long does a bond last? So for this, we did a really simple experiment. We took voles, we paired them with an opposite sex partner for 2 weeks, and then we did a partner preference test. And lo and behold, these guys have a partner preference. This is the most robust and reliable behavioral phenotype that we have ever seen, I have ever seen, and we see it in every experiment. So that's great, so we have something to study. We then separated these animals from their partner for 48 hours. We were studying only the males in this context. And then we did a partner preference test. And again, we see that they have an intact partner preference. And this is not surprising because we don't expect this acute period of separation to be sufficient to dissolve a bond. But based on sort of some literature at the time, as well as some pilot data, we separated these guys again for 4 weeks. And here we actually anticipated or hypothesized that there would be some weakening of the bond. We were really surprised when we saw this, because what this indicates is that these guys have perfectly good partner preference that remains intact even after 4 weeks of separation. Now, I do want to make the point that this is an absolutely incredible feat of social memory for a rodent. So mice and rats can typically remember other species members for hours, maybe days. And this is really unusual. They're not fighting. So what we think is going on here is we always see that. So even at this initial time point here, there's always sort of like 10% of voles that don't show a partner preference. Now, it's worth pointing out that these are not the same voles. Like maybe one of these voles in this experiment is that same vole that doesn't have a partner preference. Those are super interesting voles because the question is, why don't they bond? Are they somehow genetically predisposed not to? Did we give them a partner that they just cannot get along with, right? Super interesting questions. Hard to study when it's 1 out of 16 voles. Most of the time, these animals will show a partner preference in 2 out of 3 tests, even if they have one where they don't show a preference. One subtle aspect here is when we use the novel, they actually have exposure to 3 different novels across this time course. And we think that in some instances, the novel can be a really big factor in how they behave. And since they're not experiencing the same novel across these 3 time points, that may be explaining some of the heterogeneity in the behavior. I find that a more satisfying experience or a more satisfying answer than like voles are difficult. All right, so we were surprised by these results. Yeah, so I didn't go into that detail in the transcriptional experiment, but we only used animals that showed a partner preference. And so we know from our transcriptional data, we're looking exclusively at animals that have displayed a partner preference. And we sort of like started collecting tissue from these guys that don't show a partner preference. And then maybe eventually, if we get enough, tissue, we'll be able to run a sizable cohort that we can do RNA-seq on. This is something that we have previously leveraged, though, in our imaging studies. So, I mean, I think you know that, but we can take advantage of some of this behavioral heterogeneity to ask about how well the phenotypes we identify at the level of, say, calcium signaling track with these differences in behavior. And we've had some really cool findings within that. Okay, so partner preference is still evident even after 4 weeks of separation. And this was really surprising until we started thinking deeply about the way that we were conducting this experiment. And we realized that if you're a human and you lose someone after a certain amount of time, you have other people in your life encouraging you to move on, and you have opportunities to form new bonds. These voles did not have that. These voles were in the sort of equivalent of the castaway situation. So they're put in a cage and they live by themselves until they're given the opportunity to do a partner preference test. And if you guys have seen this movie, you actually know that Tom Hanks's bond with his fiancée does not dissolve. He actually uses it as a coping mechanism. And so we started thinking maybe social context is really important. And we know this in humans. Social support following the loss of a loved one plays a major role in how effectively you adapt to that loss. So we said, well, maybe we can recreate a scenario in which the voles actually have the opportunity to form a new bond. And so what we did is we took a whole bunch of voles here, and we went ahead and we put the opposite sex pairs together for 2 weeks. And lo and behold, they have a partner preference. You see the same heterogeneity, which is also why I like to just show the bar graphs, right? Because then nobody asks questions, but full transparency, you can see all of our animals there. We took all of these animals and we split them into 3 groups. So everybody got separated, but in the first group of animals, they got a new partner after 48 hours. They got to live with that partner for 2 weeks. Now, under sexually naive circumstances, 2 weeks produces a really robust partner preference. That's actually what you're seeing here. And in these guys, it does not. So at the group level, they do not have a preference for their partner. 2 weeks of separation revealed the same thing. So after 2 weeks, they do not form a bond with a new partner. But if we separate them for 4 weeks, now what we see is that they form a preference for this new partner. Now, one of the hallmarks of prairie vole pair bonds is that they are exclusive in nature. If you give a male 2 females and say, would you like to have a polygamous relationship? The answer is no, they will bond with one, and the one they bond with will help beat up the third vole. So the way we decided to test exclusivity is via one of the more diabolical experiments of my career, where we simply asked the males, would you like to be with your first partner or your second partner in a partner preference test? And so within this, we see that the animals that had only the 48-hour separation and did not show a partner preference for their second partner actually don't really distinguish. I mean, at the group level, they're not distinguishing between partner 1 and partner 2. The same is true for these animals at the 2-week time point, but at the 4-week time point, every single animal is preferring the second partner to the first. So our interpretation of these data is that this second pair bond is supplanting the first. And so this gives us a window, a time frame for starting to think about when we should be looking in the brain for changes. So what I've shown you in these 2 experiments is, first of all, that preference actually remains intact for at least 4 weeks under these incredibly socially impoverished conditions. But when given the opportunity to form a new bond, 4 weeks of separation is sufficient time for them to be able to now form a bond with a new partner. So if we sort of put all of this together, we take it to mean that that original bond is not fully erased, but that male voles are primed to be able to form a new bond after 4 weeks of partner separation. So something is changing in the brain that now makes it permissive for them to form a new bond. Now, I think that there's 2 key points here. So one is, if we weren't thinking carefully about social context, we would have walked away from this having not done the second experiment and come and had a very different set of conclusions. And so this just highlights the need to think about social context in the design of these types of experiments. But the other point is that this is far more translationally relevant. When we as humans lose someone, we don't forget that person. The bond doesn't fully get erased, but rather what we do is we reintegrate the meaning of that person in our life so that we're able to re-engage and form a new bond. So know that short-term separation doesn't have any real impact on their ability to display a bond, and they can't form a new bond. But after 4 weeks, long-term separation, while the partner preference might remain intact, they're able to form a new bond. And this actually is sort of the best of both worlds outcome, right? If your partner comes back, you can resume the relationship, and if not, you are at a place where you're capable of forming a new bond. So the question becomes, to enable this permissiveness. And since we had delineated sort of the transcriptional basis of pair bonding, we wanted to specifically ask the question of what happens to this transcriptional signature or gene expression signature of pair bonding. So here we employed sort of the same strategy. So I've already told you about an experiment in which we compared opposite sex pairs to same sex pairs to identify the gene expression changes that are specific to pair bonding. And here what we did is we took opposite sex separated animals and we compared them to same sex separated animals. Now, the reason that we did this is because it controls for the confounds of social isolation upon separation. So for every social mammal, humans included, social isolation is stressful and it leads to gene expression changes in the nucleus accumbens. And we're not necessarily interested in asking how does gene expression change as a function of isolation. We want to know how it changes as a function of pair bond loss. Both of these groups are experiencing isolation, but only these guys have lost their pair bonded partner. So we followed a similar schematic to what I showed you before. We paired animals with opposite sex or same sex partners for 2 weeks. We did our partner preference test, included only animals that actually showed a partner preference. We then separated them. And then we collected nucleus accumbens tissue either 48 hours after separation or 4 weeks after separation, which I'll refer to as our long-term and short-term time point. And remember that this long-term time point is also the time point at which they're capable of forming a new bond. We then compared these back to the animals I showed you previously, where the tissue was collected from animals at the same time points, but these animals remained paired with each other. So more volcano plots. So what this shows us is basically, here's the signature you've seen before, where we have some upregulated and some downregulated genes in our pair bonded animals. After 48 hours, we're still able to see some up and downregulated genes, and at 4 weeks, it's sort of the same thing. Now, the first thing we noticed is that these axes are all the same on these 3 graphs, and it seems like the size of these volcano plumes is receding a little bit. And that is a terribly non-rigorous way to sort of assess the data. But it did sort of hint to us that maybe what we should look at is whether or not there was any actual statistically significant signature of erosion across these 3 plots. So I'm just going to show you one visualization of this. Again, the paper has a bunch of different analytical approaches that all show this. But we can take our Morpheus plot that takes our most downregulated genes and then plots all the way up here to our most upregulated genes in our pair bond group. And then 48 hours later, we can keep the gene order the same and use the color to infer whether or not that particular gene is up or down regulated. Now, this is a little bit messy, and I think that reflects both that biology is messy, but also that there's a lot going on. They've just been separated from their partner. But what is striking here is that we still see this sort of like enrichment of purple at this end and enrichment of green at this end. And so mathematically, it's actually statistically, the correlation between these two is statistically significant, indicating that this pattern of expression is really similar to what we see in pair bonded animals. In other words, 48 hours is not enough time to disrupt the sort of pattern of transcription that emerges as a function of forming and maintaining a bond. At 4 weeks, there's sort of no order to this anymore. So there's no enrichment of purple on the right or green on the left. And so this basically tells us that there's an erosion of this pattern. This pattern is gone after 4 weeks of separation. Now, I'm using the term erosion in a very meaningful way because we don't think that these animals are going back to a sexually naive baseline. Bonding has left some imprint on their brain. If it hadn't, they wouldn't be able to show a partner preference. Now, we can take this one step further and we can say, okay, so we know what biological processes are actually predicted from these gene expression changes. How well do the gene expression changes in these other 2 groups predict these same biological processes? And again, at 48 hours, what we see is the sort of darkness of the blue here is an indication of how well the process is predicted. And it doesn't look any different. So again, we predict the same biological processes to be changing in the nucleus accumbens of pair bonded voles, even after 48 hours of separation. But after 4 weeks, what we see is that there's a significant reduction in the p-value indicating that these terms are much less well predicted by our changes in gene expression at 4 weeks. So again, this is sort of a biological representation of the fact that we have an erosion of these signatures. Now, as we were performing all of this, we started thinking about the fact that glia are so strongly represented and predicted within these biological processes. I think that's great. We're currently following up on, is this actually real? But we became a little bit worried that what was happening in glia might be masking what's going on within the neurons themselves. And so we wanted a way to be able to look at what's happening transcriptionally in the neurons upon loss, independent of sort of the other cell types that exist within the brain. And so for this, my graduate student made a genetic construct that allows us to perform vole translating ribosome affinity purification, or what we like to call Voletrap for short. This is a molecular technique, not to be confused with this type of Voletrap, which we use in the field. But this construct allows us to express a GFP tagged ribosome exclusively in neurons. So if we think of the brain as a series of sort of neurons and non-neuronal cell types, including glia, then what we have here is in the neurons, we have mRNAs that are being translated that have these green ribosomes. And in the glia and other non-neuronal cell types, what we have are no GFP tagged ribosomes. We can isolate all the mRNA, and then we can actually do an immunoprecipitation approach that allows us to specifically pull out these green ribosomes and their accompanying mRNAs. And so this allows us to siphon off just the portion of the mRNAs that are being made within the neurons. And so when we do this, we basically go from about 12,000 genes, which is what I've been showing you so far, to about 2,400. And so the first litmus test was sort of, if we use the same Morpheus plot, we see again that there's this erosion of a transcriptional signature within neurons. So it seems like this is true in neurons as well as glia, but we decided to take this one step further. And so we performed a clustering algorithm that allowed us to identify sort of patterns of gene expression that are particularly interesting. So I'll give you a couple of examples here. So these are genes that are downregulated, but only after long-term loss. Now, we can look at which sort of biological processes are predicted to be changing as a function of these gene expression changes, and we get really helpful terms like walking behavior. Still working on that one, but what's important here is when we look at dominant genes, it includes SHANK3. So SHANK3 is a scaffolding protein within neurons that's been extensively implicated in autism spectrum disorders. It's a genetic cause of autism, and autism spectrum disorders are characterized by deficits in social behavior. On the other end of the spectrum here, we have genes that are upregulated, but only following long-term loss. And so these two are great populations or great transcripts to be looking at as potentially sort of shifting the brain into that permissive state that only exists at after this 4-week time point. Now, the biological processes here largely include things related to sort of cellular respiration and metabolism. And so that may not seem initially intuitive, but there's actually a relatively large literature showing metabolic changes as a function of a change in behavioral state, such as a shift in hierarchy structure. And then finally, and what I think are some of the more interesting gene candidates here, we can identify genes that are upregulated upon short-term loss. Now, I want to really highlight that this is not an effect of social isolation or separation per se, because the way that we're showing the upregulation is by comparing our opposite sex separated to our same sex separated. So this is only upregulated in animals that have acutely lost a pair bond. And what's interesting here is we see a lot of terms that have been defined because they come up in the drug abuse literature. So things like response to amphetamine and amine, but what this comes down to is that the prominent genes within this category include a number of different dopaminergic receptors. And this is really gratifying because previous work that has looked at and manipulated dopamine receptors has shown that there's a really important role for dopamine in maintaining bonds via behavior over time. Today is that we've essentially been able to develop a new model that entails prolonged partner separation. And I've been able to show you that prolonged partner separation results in erosion of the pair bond transcriptional signature, and that glia are implicated in pair bonding and loss processes, and that we can do some sort of fancy transcriptional methods that allow us to look at specifically what's happening in neurons and identify subsets of genes that are likely to be involved in sort of the long-term adaptation as well as the short-term response to partner loss. Now, in an immediate sense, what we're doing is we're following up on this using single cell methods. And this is sort of serving two goals. So the first is the question of whether the cell composition of the nucleus accumbens changes. We would expect this to be the case if oligodendrogenesis is actually a really important and fundamental feature of pair bonding. So my graduate student Liza has represented this as a, little heart-based cell groups and bonded animals that are no longer heart-shaped here in the long-term animals, long-term separation animals. But this also will allow us to ask in which cell populations are we seeing the relevant changes in gene expression? So are there particular cell types that are important for bonding and or response to partner loss? So what I've shown you today is that we can sort of take what we know about the neurobiology of bonding and use that to inform the study of loss. And so in particular, we've operationalized loss via this partner separation paradigm. And then using that, we've been able to sort of delineate a change in social behavior, a trajectory of change in social behavior that gives us some insight into the question of when and whether a vole has adapted to the loss of their partner. Using that behavioral trajectory, we were then able to go into the brain and ask the question of what happens, specifically what happens to the pair bond transcriptional signature. And we see that it erodes as a function of long-term partner separation. So what does this mean? Picture context, we think that this erosion of the transcriptional signature may be fundamentally required for the animal to be able to form a new bond. And since we're using this new bond as a hallmark for adaptation in this species, the other way we can say it is that the erosion of this transcriptional signature creates a new permissive state such that these animals are able to form a new bond. Now, from a human perspective, if that's actually the case, now this gives us something to target. How can we facilitate the erosion of this transcriptional state such that individuals can become can adapt to the loss of their partner more quickly? So I've had my lab for about 5.5 years now, and I've been really fortunate because I think what we're headed towards, or what I hope we're headed towards, is an integrative understanding of bonding and loss. And so we've been doing a lot of things like the novel paradigm that we can use for studying loss in specific, as I outlined today, as well as a bunch of conceptual and technical foundation building. And my hope is that this really poises my lab to disentangle grief, loss, and more. And what I'd like to do in the last 3 minutes or so is just focus on this and more and just give you a couple of teasers for what we're working on right now. So in addition to the single cell sequencing approaches that I mentioned before, we've also become sort of really interested in two primary areas. So the first of these is a recognition that social interactions are reciprocal. So we have a tendency to study the behavior of one animal at a time in a social interaction, sort of pretending the other animal is an object. But we know that social interactions are dynamic and responsive. And so if we truly want to understand social interaction, I think we need to do it from the perspective of both animals at the same time. So my graduate student, Liza Bressman, started by asking the question of when humans form pair bonds, we also start organizing our behavior around each other. So the question is, you call someone up and you say, honey, are you making dinner or am I? Who's getting the kids, right? This is an example of organizing intra-pair behavior. So she wanted to know, do we see an organization of affiliative behavior in prairie voles that have been together for a while? So she started by pairing prairie voles and she just let them cohabitate and mate for 2 days. And then she did a partner preference test on the male and the female separately. So she took the male and he's choosing between his partner and a novel. And what you see here is basically how much time he spends with the partner. She then did it with the female. She did this interspersed. But what you see on the right in pink is how much time the female chose to be with her partner. And the gray lines that are connecting it indicate the pair's behavior. So nothing is going on at this sort of 48-hour time point. We see if we look at the data that females have a partner preference, males don't really have a robust partner preference. So she just threw them back together for another 12 days. And then she basically looked again at day 14. So these animals have been together now for 2 weeks. And previous work in my lab has shown that there's a strengthening of pair bond over that time. And here what you see is the emergence, first of all, of a pretty robust sex difference where females are huddling more with their male partner than vice versa. But then also what's really that we can also see an organization of intra-pair behavior, because reliably across these 16 or 18 animals here, what we see is that within a pair, the female is huddling with her male partner more than the male is huddling with the female. So while we have sort of what we would call high affiliation and low affiliation pairs, within a pair, we reliably see that the female is huddling more than the male. Yes. We actually looked at that. So for males, the emergence of partner preference is associated with spending less time with the novel individual at this long-term time point. So basically here, the males are spending more time with the novel than they are at this long-term time point. But for the female, it's not a change in novel-directed behavior, it's an increase in the amount of time that she spends with the partner. So I didn't go into a lot of detail here, but all of that is actually in the paper, which I should have changed from in revision to in press. It's in Genes and Brain and Behavior if we want to look it up. It's also on bioRxiv. Mm-hmm. Yeah, so that's something that we've been struggling with. So in the paper, there's actually two different paradigms that we did back to back. One is this choice test where one is tethered, and then the other is a social interaction test, freely interacting sequentially with either a novel or a partner. This is useful in that we know that one animal is making the decision about who to be with. And in the social interaction, it's really hard to tease out who's initiating the interaction. So in the latter, we still detect a partner preference. They spend a lot more time interacting with a partner than they do with the novel. But we aren't able to sort of tease out this difference. I think it's entirely possible that one of the primary drivers here is actually the stressor that the partner is experiencing, because we know that these guys will engage in that sort of consolation behavior. Regardless, I still think it's very striking that we see this robust and reliable sort of, so we can actually like calculate the likelihood, like the percentage increase that the female will display affiliative behavior over her male partner. Also, what I find really interesting, and this is digressing just a little bit, these are intact animals and only one pair did not get pregnant, and it's the only pair in which the female does not huddle more than her male partner. Okay, so this shows us that there's some organization of behavior as well as an emergence of a sex difference as a function of bond maturation. The question is, what's going on in the brain that may underlie this sort of change in social behavior? And for this, we realized that we needed a way to be able to look at the brains of more than one individual at a time. And so as a first pass, we've been using fiber photometry, which is just a technique that coupled with various fluorescent sensors can give you real-time readout of something that's happening in the brain. And so the first of these sensors is actually one that lets us measure real-time dopamine release within the nucleus accumbens. And so that's what you're seeing in this sort of spiky trace here. And so this is my graduate student, Ann Pierce, who was able to actually record from prairie vole pairs as they were mating. And so in the middle here, what you see is the behavior. And if you've never watched rodents mate, basically it consists of bouts of mounting and intermission by the male. Then he gets off and it's like wash, rinse, repeat. So here, the black bars indicate when the male is mounting the female, and this indicates the levels of dopamine in the female nucleus accumbens and in the male nucleus accumbens. And what I find quite striking here is that you actually see the biggest increases in dopamine immediately after the male dismounts from the female. And this is not true for the male. So that's something we can talk about over drinks later and always makes for great fodder. This is very much a just so story at this point, although there is evidence in rats that female dopamine levels increase after mating. The other way that we've been approaching this is to use a fluorescent calcium sensor that allows us to sort of get a sense of the bulk sort of neuronal activity within the prefrontal cortex. And so one of my other graduate students, Kathleen Murphy, has been taking prairie voles, and the pink trace is the female and the blue trace is the male. When she puts a divider between them, what you see is that these traces look pretty dissimilar. But when she takes the divider out and they're interacting with each other, the actual traces themselves, the oscillations of the activity actually start becoming much more similar. And this is something that we can quantify a number of different ways. What I'm showing you here is just the Pearson correlation coefficient between these two traces. And what she has shown is that in animals that have formed a bond, they have like tighter correlation with their partner than they do when they're interacting with a novel individual. And so we think this serves as almost like a proxy for how you can look at your partner and know what they're thinking, which you can't necessarily do with someone that you don't know. Yes, I realize that's totally anthropomorphic and whatnot, but I think it gives us a handle for thinking about what might be going on. The other area that we're moving into, or sort of continuing to work in is harnessing species diversity. And so we're doing this at 2 levels. Within my lab, my graduate or my postdoc, Lisa Hura, is comparing prairie voles and their promiscuous cousin, the meadow vole. And so she's testing the hypothesis that differences in dopaminergic reward mesolimbic pathways is responsible for differences in bond formation. And she's using a number of the tools that we've sort of already talked about. Then in a much more extended sort of species comparison project, we're working with UCLA and Berkeley to compare mice, voles, and bats, which represent a spectrum of different social behaviors, as well as UCLA miniscopes to ask how the encoding of a mating partner differs as a function of your social structure. And so what I've shown you today is that we've been able to delineate some of the molecular changes that are likely to underpin pair bonding, and that we've been able to leverage what we know about pair bonding in order to gain some insight in what happens during loss and loss adaptation. And then finally, just a couple of teaser slides at the very end. But I would be remiss if I didn't point out that there is a huge number of people, and I have been incredibly fortunate to be able to recruit fantastic trainees, many of whom have now gone on to graduate school. And my first PhD student is going to graduate this spring, which is super exciting. I've also been really fortunate because none of this would happen without the support of both private foundations as well as NSF and NIH. And then finally, I'll just make a quick plug that I am indeed looking for a postdoc to work on sequencing, activity-dependent labeling, and more. The notice of award from our R01 came in last week. So thank you so much. Yeah, so the question was whether or not we've performed the same transcriptional experiments in females, because the data that I showed today was exclusively from males. And the answer is very simple. It's no. That's aim 1 of the R01. So the question is, are there sex differences? I would not be surprised if there are. We know that behaviorally there are sex differences, not just what I showed there, but also that females tend to bond more quickly than their male counterparts. That's a great question. I think one of the really big challenges here is that all of the manipulations we do are things that we can sort of test once per animal, right? So we can run a time course, but we can't necessarily test the same animal over and over again. So if our metric is when can you form a new bond, that's something we can test essentially once. If our metric is how is your gene expression changed, then we can test that metric once when we take the brain. And so these are all really interesting questions related to what are the external factors that may contribute to how you adapt to loss. And it's certainly something I'm super interested in following up on. We can sort of draw a bit from the human literature. So one thing we know is that people who actively avoid engaging in grief actually have poorer grieving outcomes. So that suggests that it's not just time passing, but we're actually actively engaging with the loss and moving on from there. Okay, so the question, if I try to paraphrase it, is essentially that it's totally different to live with a partner than it is to be by yourself. And so maybe partly these gene expression changes that are a function of partner loss are actually just reflecting a difference in sort of how you live your daily life or the behaviors that you're engaging in. I feel like you were like a reviewer on the R01 and you were very dissatisfied at our focus on the nucleus accumbens. Yeah, so I think that there are brain-wide changes, right? And that I highlighted one fMRI study that sort of highlighted the role of the nucleus accumbens, but there's actually an emerging literature suggesting that prefrontal sort of mesolimbic loops, thalamic loops are involved in sort of ruminating and perseverative grief and things like that. So I don't think that the nucleus accumbens is like the seat of grief. So there's sort of 2 questions here. One is what is going on in the rest of the brain? Thank goodness I have an entire career ahead of me. And that's about all I can say at this point, but we are interested in it. Where we're branching out now is mostly the hippocampus and the prefrontal cortex. And that's because of the role of the hippocampus in social memory and the role of the prefrontal cortex in sort of overlaying sort of a contextual basis to what's going on. We think the nucleus accumbens is the seat of that reward association for pair bonding. The other question is sort of, maybe I can partially answer it by saying that I think one thing we can do, and we're trying to do this experiment now, is ask, is the transcriptional signature of your second bond similar to the transcriptional signature of your first bond. Because to me, that says basically like you have two states. You have an unbonded state and you have a bonded state. And in one unbonded state, you might be sexually naive, but in another one, it may just be that you have lost your partner and you're adapting to that loss. I don't think from there we can necessarily tie that back to specific differences in behavior, but it's not surprising if there will be differences in behavior, and I would not be surprised if that's one of the mechanisms that's contributing to these transcriptional differences. I think it's potentially impossible to sort of uncouple these two, right? Like, how do you uncouple the behavioral change that accompanies loss from the transcriptional change that accompanies loss? It's not that clear cut because in the head-to-head test, they're not reliably choosing the first partner. So it's almost like they're in this weird in-between land. Okay. So the one thing we've been reliably able to show is changes in anxiety that are more robust in animals that are separated from an opposite sex partner compared to those that are separated from a same sex partner. And so we've shown this with elevated plasmase, with looming stim, which is basically where you pretend there's a predator coming at the vole and they have to run away, or they do run away. How quickly do they run away? Sorry, the question for the Zoom audience, the question was, do we think these voles are actually grieving? This is a really hard question. So we can measure all these things that are similar to what we see in grieving humans, like activation of the HPA axis, increased anxiety levels, increased stress responsivity, changes in pain thresholds. Like, all of these things seem to replicate in the voles. My personal take on this, my bias as a scientist, is that all of these are a response to trauma, and that we see similar things happen and following other traumatic events. And that what we really need to be homing in on is the specific selective social motivation that underpins yearning in humans and whatever its sort of equivalency in a vole might be. So maybe in a couple years, if you invite me back, I will show you some work we're doing in which we're having voles lever press to gain access to their partner, and they will do so robustly. And so the question becomes, does absence make the heart grow fonder? Does it matter if it's been 2 days or 4 weeks? And so those specific loss reaction experiments are underway currently. Right, so the question is, there are a number of pair bonding bird species as well. Probably the most well-studied is zebra finches, and their response to partner loss, or the sort of point at which they're capable of forming a new partnership, is much faster than what I've shown you here. So I would highlight, first of all, I would highlight some species differences that exist even with respect to bonds. So in zebra finches, if you have arranged marriages, it produces far fewer offspring than if you let those zebra finches pick their partner. So there are aspects to the way that voles pair bond and birds pair bond that are actually different. The partner loss. So the short long answer is that these guys seem to undergo a rebound effect. So I didn't show data, but if we do a partner preference test sort of right after we give them a new partner, all of them show a partner preference. But we almost think this is like they're so excited that there's something new and they don't have to live by themselves that they associate the animal with that. But under normal conditions, after 2 weeks, we see an increase in that partner preference. And in both the 48-hour and 2-week group, we see a waning of that partner preference, which I think is really interesting. I don't know, that's why it's like, that slide title is like rebond or rebound, is in reference to that, even though I didn't show you all of that data. That's really cool. I didn't know that. So I will say, for the people on Zoom, basically, the revelation is that zebra finches will go back to their first partner, even if they haven't seen them for like years. So I feel really bad for those second partners. And that's clearly not the case for the voles, right? So when we did that exact test, if it's been long enough, they prefer their second partner. Yeah. So the question is, how does the reproductive physiology of voles sort of fit within our findings? And I think so first, what I would highlight is that they've done field studies where they've asked, so when they pair bond, they have a burrow, they form a territory. So if one animal loses their partner, how long does the other animal stay at the burrow before they disperse and presumably look for alternative mating opportunities? And the answer is on average 17.5 days, which fits nicely between that 2-week and 4-week time point. The other part of your question, though, is, how does this fit reproduction wise? So prairie voles have a 3-week gestation. So if you don't have any pups around 3 weeks after your partner died, which, not surprising, but like there might be sort of a biological set point that says, absolutely, there's no possible way that you are producing offspring with that individual. So it's time to move on. So I do think that the reproductive aspect of this is an important part of the timing. In humans, 4 weeks is not. When people see like dramatic changes with respect to grief, it's more like 6 months on average. We also live for much longer and have much longer gestational cycles. So yeah, so the question was, how well would some of these findings generalize to other types of attachment relationships? So for instance, we get asked a lot about how this might relate to parent-offspring attachment. So one thing that's really nice about pair bonds is that they are stable over the long term. When we start talking about parent-offspring attachment relationships in rodents, first of all, we can ask some questions about the specificity of that attachment, especially during the early stages where moms will just take care of any pup. But even more important than that is basically that for rodents, it is normal for the pups to sort of grow up, and 3 weeks later we wean them in the lab or in the wild. They might go off and disperse to find their own burrow and mating partner, etc. So these sorts of longitudinal studies looking at loss are actually really tricky to do in that context. However, having said that, I do think that the basic tenets are going to be sort of broadly conserved across different attachment types. And so the best evidence for this is actually the work on oxytocin, right, which has been widely implicated in attachment relationships between parents and offspring, as well as pair bonds, as well as friendships. And so I think there are some differences across those relationship types, but the general framework is going to be biologically applicable across them. Great questions. Thank you so much.
Thank you.